It is because of pneumonia, and further we have many kind of problem in Korea, due to which many deaths happen. So still cancer is a big topic, even after coronavirus infection or corona pandemic. So we are also working on various topics related to cancers. So first of all, I want to tell you about basic mechanism of how cancer's immune recognition system work. Basically, cancer have two kind of problem. They have low immunogenicity, and they used to suppress nearby immune cells by themselves by releasing various kind of chemokines, cytokines, or various other growth factors. So low immunogenicity means cancer cell can be detected by immune cell, but they cannot kill them. So for this kind of research, in 2018, Nobel Prize is given to two people, one from USA, one from Japan. That research was related to CTLA4 and PDL1 receptor. So this is the main problem they also try to solve, and we are also working how we can increase the immunogenicity of tumor cells. So immune cell can recognize them as a cancer and can kill them immediately. Second problem is immune suppression. Immune suppression is basically by cancer cell try to release many kind of factors by which this immune cells cannot try to attack them. So these kind of factors also we are trying to remove by using plasma. And whenever we talk about the immunomodulations, especially in cancer case, we have to think about cancer immunity cycle. And those two people who got Nobel Prize in 2018 were James P. Allison and Tosuku Honjo. These two professors worked on immunity cycle. This is the immunity cycle I am going to describe you. This immunity cycle has 7 stages: stage 1, stage 2, stage 3, 4, 5, 6, and 7. In first stage, they explained that release of antigen by treatment. Treatment can be anything. It can be chemotherapy, radiation therapy, or some targeted therapy, or it can be PDT. Nowadays, PDT is also very well-known therapy. And it can be plasma also. It can be any other therapy, by laser or by light, anything. It can be anything. So at first step, there is always a release of cancer antigens by treatment or by themselves. After this release of this cancer antigen, they are recognized by dendritic cell. We also say them APCs, antigen presenting cells. They take those antigens and present this antigen to other immune cells. So that's why we say them APCs, also antigen presenting cells. Usually they present this antigen to T cells and they activate T cells. These activated T cell goes to the blood vessels towards the tumor site through blood vessel, and at 5th step they try to move from the blood vessel to the tumor tissue, and at 6th step they try to recognize T cells, cancer cells by T cells, and at last step, that is 7th step, that is killing step, you can say, killing of cancer cells by this trained immune cell. So this is the whole immunity cycle. If anyone working on the immunomodulations, these 7 steps are enough. No need to go here and there. Just focus on this 7 step. Do something for this 7 step, and then you can get very wonderful results. So we are also focusing on first and second and third, those all steps in plasma medicine areas nowadays. So these are some plasma related applications we are doing at PBRC. So by using plasma, we are trying to increase immunogenic cancer cell death in tumor. We are trying to increase the immunogenicity. And in second application, we are working on stimulation of immune cells. Those are white blood cells, like macrophages, like monocytes or other immune cells. So we say direct immune stimulation of immune cell. And in third area, we are working on neutralization of immune suppression. So whatever immune suppression created by the tumor tissue, we can neutralize it, we can stop it by using plasma. And the final goal is preparation of cancer-related vaccines by using plasmas. So all these 4 areas are related to each other. I'm telling you one thing, by just one device, you can solve all these 4 areas. So this is one study where we worked on the direct effect on immune cells. Here we taken macrophages. Macrophages are wonderful immune cells, which can be very helpful to kill bacterial infection, to eradicate bacterial infection or viral infection, as well as they have very wonderful role in cancer treatment. So in this case, we taken macrophages and we tried to treat them by the plasma, and we found wonderful result. These macrophages are well differentiated after plasma treatment. Previously, they were in resting stage. We taken resting stage macrophages and differentiated with plasma. And we also checked their motility, and we did motility analysis, such as we checked migration and their invasion capacity, how much they have a power to move by using migration and invasion assays. That is also enhanced after treatment. We also checked various other experiments, like whether these treated macrophages are killed by plasma treatment itself, or they have some problem or some biocompatibility. And we found there is no such toxicity by this plasma on the immune cells. And after activating these macrophages, these are the facts data for those macrophages. These activated macrophages, we grown together with cancer cells. We taken 2 kinds of cancer cells. One is lung cancer cell, A549. Another is T98G, that is brain cancer cells. We co-culture this brain cancer and lung cancer cells together with these activated macrophages. And we found that the death in this co-culture condition, death in the cancer cases is increasing. So it is directly related to effect by the plasma treated macrophages. And we also found the mechanism that the mainly ionous inducible nitric oxide synthetase expression is increased, as well as TNF alpha, that is called tumor necrosis factor, that is also enhanced in this co-culture condition. It means these 2 molecules are important in mechanism case for killing of this cancer cells under co-culture condition. In another study, we activated monocyte, blood monocyte. We taken blood monocyte from patients, and then we activated them by plasma and waited for 7 days to 14 days for their activation. And later we co-culture with the cancer cells, and we found anti-cancer, very wonderful anti-cancer activity. Here you can see, first we check the differentiation of this monocyte by plasma. We found wonderful effect. Differentiation is very high. And we check the marker, because macrophage can be of many types. It can be M1 macrophages, M2 macrophages, M0 macrophages. So every macrophage have their different application. So in our study, we wanted to check what kind of macrophages these are after activation. And we found that these macrophages, basically anti-cancer macrophages, M1 macrophages rather than M2 macrophages. First, we use 2 kinds of marker for this study, CD86 positive and CD163 positive populations we checked. And these are 2 CD markers which are present on all macrophages, but their ratio is different in different macrophages. Like in M1 macrophages, you have more CD86 positive cells, or in M2 macrophages, you can have more expression of CD163. In our population, we also find when we treated these macrophages by plasma, these differentiated macrophages are more towards M1 macrophages, more expression of CD86 rather than CD163. So in this experiment, we found that whatever we are differentiating or stimulating, or you can say activating, that is more better for cancer therapy rather than any other application in this case. We also checked whether these activated macrophages are releasing some anti-cancer cytokine or chemokine or something else. And we found that the several cytokines who are related to anti-cancer activity are enhanced in this experiment. We checked by microarray assay also, and as well as by PCR data, expression data. And we also checked whether these activated macrophages by plasma can kill cancer or not. So we co-culture these activated macrophages together with cancer cells. We taken two kind of cancer cell here. One is U251mg and another is U87mg. These both are glioma, both are brain cancer cells. We co-culture these both brain cancer cells together with activated macrophages. And we found wonderful effect. We found that when we co-culture these cells together, there is a decrease in the cancer growth in this co-culture condition with activated macrophages. In both cell line, we got similar kind of data. And more wonderful effect, we got that cell, whenever we co-culturing with the cell, we have more effect rather than supernatant. Even supernatant of this activated macrophages is killing cancer cells. That is also a very important data. In another study, we are focused on the DAMP, ICD and DAMP. First of all, many people maybe don't know about ICD and DAMP. First, I will describe about what is ICD, what is DAMP. ICD is immunogenic cell death. Basically, whenever we treat cancer, any cancer cell by any treatment or any stress we give to cancer cell, they used to release some molecules. And these molecules can be recognized by immune cells, and then immune cell can be activated against these cells. So this is the process of ICD, or you can say DAMP release. So whenever a cancer cell have any stress, they try to release HMGB1. This is nuclear factor, homeobox protein 1. And they also try to release ATP from cytoplasm to outside environment. And they also try to release calreticulin, and they try to present this calreticulin on the surface of cancer cells. And also they try to release many kind of interleukins and chemokines, which are responsible for immune activation. So we wanted to check whether our plasma can do this kind of DAMP release. And DAMP is damage associated molecular pattern. These are molecules which are responsible for immune response against cancer. And in our study, we focused on ATP release that we called as find me signal, find me for cancer. And CRT is eat me signal. This is the signal for immune cell to eat these cancer cells. And HMGB1, HSP90 are another signals which are important for any immunomodulation study in cancer immune system or immune biology. So we checked, we tried our DVD plasma on various kind of cancer. This is the some very specific data. We checked ATP released by the cells after treatment. We found the high level of ATP is released when we are increasing the plasma exposure dose. So in both cases, you can say that here, this is the plasma of air. This is plasma by the nitrogen gas. In both kind of gases, plasma ATP is increasing. It is very significant data to show that yes, plasma can induce DAMP release in tumor cases. We also checked HMGB1, that is homeobox protein 1, S100, that is also nuclear protein, calreticulin expression on the membrane, and we found in every cases the DAMP is releasing after plasma treatment in significant amount. We also checked by PCR assays various kind of DAMP molecule, such as calreticulin, HMGB1, IL1, HSP90, HSP70 and 90, and TLR, that is toll-like receptor, that is also related to DAMP. And we found a significant increase of TLR, IL1, HMGB1 by air DBD plasma. And in nitrogen cases, there is a variation between nitrogen and air gas plasmas. In nitrogen cases, we got calreticulin and HSP90 is more expressed as compared to air gas. And finally, we compared in our studies that whether plasma is really good in medical areas or not. So we compared with radiation therapy, and we can compare it with the photodynamic therapy, that is PDT. And this is our table. You can see here. This is plasma, non-thermal plasma. Another example here is RT, radiation treatment, and another one is PDT, photodynamic therapy. So if you see here, Everything is okay. Target, invasiveness, or mechanism of action, depth of effect, caused by ICD. But these all things are okay, but two kind of factors like side effects and direct effect on the immune cell are very variable in case of radiation therapy and PDT. They are differently behaving in those cases. So if you see in non-thermal plasma case side effect, there is no major side effect reported yet. So we are also studying our collaborative group in USA and Europe also studying these plasmas for side effect. So up to now, there is no major side effect by plasma to the human being. However, in radiation therapy, skin changes, second cancer, second site specific tumor occurrence, and damage to the nearby organ are well reported. As well as in PDT cases, some skin rashes and skin changes are reported. And in case of direct effect on the immune cell, this non-thermal plasmas have preservatory functions or stimulatory function or activation function that I showed you in today's presentation, that plasma can activate immune cells such as macrophages, monocytes, or even they can induce immunogenicity in cancer cells by release of DAMP or inducing ICD. And in case of RT, radiation therapy, this activation of immune cell is suppressive in nature. So there is no activation of immune cell. Rather activation, they are suppressing these immune cells. And same case in PDT therapy also, because in PDT we use several kind of chemo, photosensitizers and laser light source that have some kind of suppressive effect on even on immune cell. That is reported thing, not by ourselves. So we can say plasma can be safely used in future in all medical areas, including cancer treatment. Now about plasma nanomaterials. Plasma is well known for synthesis of nanomaterial. It is already reported by previously by many researchers. Basically hot plasma. Hot plasma means temperature is very high. Plasma temperature is very high in case of hot plasma. You cannot touch hot plasmas by your hand. So there are several examples of hot plasma. I will not explain you here, but hot plasmas are many times used for the preparation of nanoparticle, not cold plasma. So we tried to use cold plasma at PBRC, and our collaborators also tried to use cold plasma in PBRC for synthesis of nanomaterials. So we did it well. We are working on it. These topics are nowadays also. And we are also working on the plasma plus nanomaterial, like synergistic effect of plasma and nanomaterial for specific biomedical application. So we are doing it at PBRC. In one study, we reported that when we treat any cells for delivery of nanomaterial or nanodrugs or nanomaterials, plasma helped them to go inside. How? We reported that the plasma can induce leaky membrane, which is responsible for more uptake of drug or more uptake of nanomaterial inside the cell. So this work is already reported. And in another study, we checked whether this plasma synthesized nanomaterial, because these are green synthesized nanomaterial, no harsh chemicals are used for the preparation of nanomaterial in plasma-based nanomaterial synthesis. So we checked whether these nanomaterial are different than the market available nanomaterials. We checked two kind of nanomaterials here. One is gold nanoparticles available in Merck, sigma rich as control. And another nanomaterial we prepared by ourselves at PBRC. And we checked effect of both nanomaterial for immunomodulation. And we found our nanomaterial, which is synthesized just by plasma without having any harsh chemical, these are working well. They are inducing more immunogenicity in cancer cell. We checked in breast cancer cell in this study. And also they are inducing autophagy. That is also a wonderful process for cycling of materials of cells. So we got very wonderful result by our nanomaterial as compared to the Merck market available nanomaterial. And we found this effect is because of that those nanomaterial are synthesized by some kind of acid or bases, which can be harsh to the cells and which can be toxic to the normal cells or cannot have proper biological applications. And this is the mechanism how we can use only plasma without using harsh chemical. Even in presence of water, we can synthesize nanomaterials. So this is about the reduction process. Reduction process is by the electrons, solvated electron, which are produced by the plasma in the media. Media can be water, media can be buffer. So basically these electrons, solvated electrons, are helpful for synthesis of nanomaterials. And we checked it in gold nanoparticle case, silver nanoparticle. Nowadays we are trying to iron also. And we are also trying to the carbon-based nanomaterials. We are also trying to make hybrid nanoparticle by our some specific methodology. And these all nanomaterial synthesis are one part synthesis. There is no need of like many steps. One step process and green in nature. And not only preparation of nanomaterial, we can also functionalize nanomaterial by using plasma. So because plasma have many kind of ROS, reactive species, and some examples are mentioned here. These are some examples. They have a lot of reactive species. One species is hydrogen peroxide, S2O2. Another is hydroxy radical, OH negative. And this is superoxide. So these are few example of reactive species which can be helpful for functionalization of nanomaterials. You need just polymer, you need some biomaterial, whatever you want to functionalize on your nanomaterial, and no need of any acid, harsh chemical, or any base for even for synthesis, even for functionalization. And these nanomaterials are working well for biological application with having very less toxicity to the normal cells. So this is our one study in which we use this plasma prepared nanoparticles for brain cancer, reduction of brain cancer or inhibition of brain cancer. We reported mechanism together with the nanoparticle. We reported here uptake by the TEM, as well as we reported the effect on the in vivo models, the reduction of the growth of the tumor, and effect on the metastasis. That is, for that we measured EMT, many kind of EMT markers, as well as we checked the cancer stemness, whether we can reduce cancer stemness by using this kind of nanoparticle or not, by using synergistic effect of plasma and the gold nanoparticles. So we found wonderful result. Yes, we can reduce stemness. We can also control EMT process in cancers by using this biocompatible nanomaterial and plasma. Another study we checked we did on gold nanoparticle, that is also related to immunomodulation. And in this case, we use two kind of cancer cells, MCF7, that is breast cancer cell, and MGA MB231. These both are breast cancer cells. And we checked a few markers of DAMP, damage-associated molecular patterns, such as CRT, calreticulin, and HMGB1, that is homeobox protein 1. And we checked the expression of, after this treatment, and we found wonderful effect, increase in the expression of both HMGB1 and CRT. Also, we checked by PCR analysis the several DAMPs, including CRT, HMGB1, S100, ATP, HSP70, HSP90. We found DAMP molecule HMGB1 is significantly enhanced, 4 times enhanced as compared to other DAMP molecule in this study. And that we also checked this DAMP molecule, whether this DAMP molecule is cellular DAMP or it is really released by the cell. So we checked two kind of DAMP molecule here, CHMGB1 and SHMGB1, and we found the SHMGB1 is more enhanced in this treatment cases rather than CHMG1. It totally give us clue that HMGB1 is released by the cell, outside the cell, because it is in soluble form in media, including CRT. CRT expression is also slightly enhanced in this population. So this is one result by using biocompatible plasma synthesized green process nanomaterial for immunomodulation purposes. And likewise, we did several kind of studies with various kind of cancer cells like glioblastoma, melanoma, breast cancer, colorectal, lung cancer, with various kind of plasma devices such as plasma jet, surface DBD plasmas, or various other plasma with various nanoparticles such as gold nanoparticle. Gold nanoparticle are very famous in this field, and other iron nanoparticle or other biological nanoparticles we used for many studies up to now. In another study, we are in another area, we are focused on virus inactivation by using plasma. Due to pandemic, many people started work on viruses. We also started in 2020, virus inactivation effects by plasma. But before that, many group in worldwide who are working on viruses, they already reported many publications by plasma, not on coronaviruses, but they published on various other DNA viruses or RNA viruses before. So it is not new that by using plasma we can kill viruses. This is old history. But we also started this work at PBRC after this pandemic, and it was the need of that time. So basically, by plasma, we can treat various kind of viruses. And plasma mechanism for virus inactivation is based on reactive oxygen species, reactive nitrogen species, UV radiations, ions, and electrons. And in this mechanism, plasma try to disturb nucleic acid, damage nucleic acid, and proteins of virus. And after degrading this virus protein and other things, they try to inactivate virus. So only this is the very simple mechanism we have by using plasma. And at PBRC, we are working on various areas in virus inactivation. One area is virulence factor. Virulence factor, as you remember, is spike protein in coronaviruses. We have a wonderful effect. We can reduce this virulence factor like spike protein by using plasma. And in the mechanism, it can be oxidation or functional impairment. And another area in virus inactivation, we are checking whether this virus, whether our treatment can alter virus binding or entry to the human host cells. And another area in this case is host immune response after this treatment. So we are working on multiple topics: virulence factor, after cold plasma treatment, virus binding and entry after cold plasma treatment, and host cell response, specifically immune response after cold plasma treatment. So this is one example for surface sterilization. This is not for treatment. This is for surface sterilization. We checked whether this plasma can sterilize surfaces or not. So we checked on plastics, metals, cardboards, and footballs, basketball, baseball. And we cultured coronaviruses on these surfaces and immediately treated by plasma and checked their growth inside host cells. And we found in all cases, this plasma is reducing coronaviruses significantly. So it means we are disinfecting these surfaces. We are inactivating viruses significantly after this plasma treatment to these various surfaces. And recently we invented one new technology. We made a very wonderful plasma device for making large amount of NO water. NO water means this species is specific species. You already heard about nitric oxide. So this is nitric oxide water. In this machine, we are specifically making gaseous NO, and which can be immersed in the water, and we can make a stable. Important thing is here, stable NO water for long time. And we can, if we are storing this NO water in -80 degree in very cool temperature, then we can keep this NO water stability up to several years, even several years. So this is already reported by us in good journal recently. So basically by using this plasma device, we are making NO with the help of nitrogen, which is present in ambient air, and oxygen that is also present in ambient air. And we are using oxygen cylinder also for supplying this gas to this machine. And specifically by using plasma, we are making gaseous nitric oxide, which can be added to the water and can be NO plasma activated water can be prepared. This NO water can be used easily for virus inactivation. And wonderful thing is that we can drink, it is drinkable. So even you can drink it without having any big side effect. That we already checked and reported in very nice journal, that is Bioactive Materials in this year. So by standardization of this device, we obtained up to 4,000 micromole of NO inside NO water. So this is very high concentration. We can store it, we can dilute it, and we can use it for further our experiments or further treatment applications or whatever. In this case, we check this NO water effect on the coronaviruses. So whenever you want to check any virus inactivation of products, you have to measure two kind of things. One is cytotoxicity of that product on the human host cells. So we checked cytotoxicity that we called CC50, cytotoxic concentration 50. So we checked the NO water cytotoxic concentration 50 together with the positive controls such as market available hydroxychloroquine. This is also famous malarial drug, which is used, some people used for coronavirus treatment. It was true. That is, it can inhibit coronaviruses easily. And another drug was ritonavir. Ritonavir was also famous drug like remdesivir for virus inactivation. And we found here the cytotoxic concentration of NO water was very high. It means it is not very toxic at below this 2000 micromole concentration. So this is very wonderful results we have. And if you see the positive control, you can see even at 100 micromole, it is killing more than 50% cells. And the same problem in the ritonavir drug. This is also a little bit toxic. And later we found cytotoxic concentration is okay. So main important thing is that whether this product can really effectively kill virus or not. For that, we used to measure EC50. EC50 is effective concentration 50, by which you can kill 50% viruses by your product. So we checked NO water EC50, chloroquine EC50, ritonavir EC50, and we found the EC50 of this NO water is also very nice. After these two concentration analysis, CC50 and EC50, we can calculate therapeutic index. Therapeutic index is what is therapeutic index? In therapeutic index is SI index, we called in virus biology. SI is selectivity index of drug or selectivity index for any treatment. How we can obtain it? We can divide CC50 by EC50 and we can get therapeutic index or selectivity index of all antiviral product. So in this case, you can see 3 product selectivity index. NO water selectivity index is 33.8. Hydroxychloroquine therapeutic index is 11.5. Ritonavir selectivity index is 6.2. So you can see ritonavir and chloroquine are very near to each other. Many people claimed hydroxychloroquine that it is not perfect drug, yes, because of its toxicity. It is really toxic to the human cells. So even though it is effective against virus, but it is toxic to the human cell. So not recommended always to treat coronavirus or any virus infections by this kind of drug. But if you see the NO water selectivity index, this is very high. In many studies, remdesivir drug have similar therapeutic index or similar selectivity index. So we can compare and our NO water simply produced cheap cost without having any much money, and we can supply it in very large amount, and therapeutic index is 33.8. So this paper is published in Bioactive Material Journal. Its impact factor is around 17 and published within this year. We also checked various kind of gene. Everyone used to do it whenever they go for virus inactivation. They used to check N gene. Many students know about it. N gene, E gene, M gene. N gene means nuclear genes, nuclear related protein gene. Envelope related protein gene is E gene. Membrane related protein gene is M gene. So we also checked in our study, we checked on the 3 concentration, 50 micromole, 100 micromole, 200 micromole concentration of NO water, together with positive control drugs, hydroxychloroquine and ritonavir. In some cases, we found our NO water is working more better than this market available drug. And we also checked genes for human host cells. And in this case, we used lung host cells. We checked whether this treatment enhancing defense capability of our human lung cells or not. And we checked a few kind of genes such as ABCC9, BIRC3, DDIT4, FOSL1, IRF1, IRF2, and CD13. This is surface marker CD13. We checked all these genes and found NO water is working well. It is not only reducing virus, it is not only inactivating virus, it is also increasing the host capability, the capability, host defense capability against virus. So we can have dual kind of effect, virus inactivation or increasing the host defense system for inhibiting these viruses. We also check the spike protein. So spike protein, you can see here, this is cell control. Human cell don't have spike, so you cannot see any spike in this group, cell control. Another is virus control, in which you can see red color around nucleus. Nucleus is blue color here. So this red color represents spike because we used antibody, specific antibody for spike protein. And that is this in human host cells. You can see a lot of, in cytoplasm, a lot of red color. That is because of spike protein. So this is virus control, in which we infected cells with virus. And another group is where we treated these cells by NO water. And another group is market available drug treatment. We found that the our NO water at 100 micromole, non-toxic dose can decrease almost 90% viruses easily. So you can see the graphical data here. That by NO water, this is spike protein is producing too much. Even it is reducing by hydroxychloroquine, but it is toxic. This is FACS data. You can see in FACS data also, it is reducing too much, up to 90% reduction we obtained. And later we reported the mechanism, whole mechanism also. Like this NO water, not only it reduced the spike protein, it also reduce genes of N, M, and E related to this RNA virus. And this NO water not only reduce gene of virus, it also increase the capability of host defense to fight against these viruses. In another study, we used not plasma treated water, we used ozone, plasma generated ozone for inactivation of viruses on surfaces. This is not for treatment. This is for the surface treatment, like in classrooms, in hospitals, we want to sterilize surfaces where many patients of virus infections were under treatments. So those kind of things we can do. Even in schools, colleges, we cannot control people, number of people. Those places we can use this kind of devices to sterilize surface viruses. Even in public transportation, we can do this kind of, we can use this kind of devices for inactivation of viruses. So in this case also, we check the selectivity index of plasma generated ozone, which is 6.51, more better than the market available drug. It is similar or more better than the market available drug. So we can just use plasma device in classroom, in offices, public departments, transport, or Wherever you have many people, you can use those devices like function, auditorium to kill viruses immediately on the surfaces to control virus infections during this kind of pandemics. In another study, I want to tell you here one more thing. Maybe many people know about the INMAS. INMAS is the Institute of DRDO, Defense Research Development Organization. INMAS developed, said that 2DG, 2-deoxy-D-glucose is the drug that they invented, and this is perfectly working well on the coronavirus patient. But it was not true. It is not invented by INMAS. Parallelly, many institutes working on 2DG, and 2DG is market available drug you can buy anytime. Nobody invented it. It is more than 60-year-old drug, which is available in market. You can buy and you can use it. So we are also studied on 2DG, effect of 2DG on the virus inactivation. So in this case, we are controlling, using 2DG, we are controlling metabolically, because we are altering the metabolism of host cells for coronavirus inactivation. And we found very wonderful result by using 2DG. So almost more than 4 log order, you can say 99.99% virus we can reduce by using plasma plus 2DG. So that is also report by us last year. And nowadays you heard about new mutants, like in last 2 years, we have many mutated viruses of coronaviruses. One mutated virus was D614G. This D614G is because of mutation in spike protein. So we are targeting those mutations, whether we can control those mutations in coronaviruses, or whether we can target mutated future mutating emerging viruses in future by using plasma. Another virus this year was monkeypox virus. So monkeypox virus infection is not by the spike protein, it is by the EFC complex. EFC means entry fusion complex. So EFC complex is made up of many proteins, not single spike protein. It is a group of proteins. More than 12 proteins are involved in EFC complex, entry fusion complex, we say. This is the DNA virus, not RNA virus, but still we are trying to obtain a strategy to control this virus by plasma. So this work is ongoing on monkeypox virus. We are controlling entry fusion complex, EFC complex, which is responsible for entry of this monkeypox virus in the human host cells. So we are targeting by plasma. This research is undergoing. Result will be shared later on. Not only viruses, we are also working on bacterial inactivation by using plasmas. In this case, we use some viruses which are present in air, soil, and water. So these are basic bacteria, not viruses, but basic bacteria like E. coli, gram-positive and gram-negative bacteria we taken in the study, like P. aeruginosa and S. aureus. These bacteria we treated by plasma, and we got some log order differences after treatment of this NO water. So this NO water is having wonderful role. Not only it is inactivating virus, not only is it is controlling cancer cell, it is also killing bacteria, different kind of bacteria. And we checked many kind of assays on this antibacterial activity of NO water at various doses. In another case also, we checked that NO water, what is the mechanism by which NO water is killing bacteria. And we found that the nitrosative and oxidative stress regulatory markers are enhanced after treatment of NO water to the bacteria. And because of these two markers, bacteria killing is happening. And we are also working on various, not only NO water, we are working on like plasma treatment to the water, simple water, buffers. And there is a very wonderful solution which is used in medicine always, that is ringer lactate buffer. Ringer lactate buffer is, so whenever any person have accident or something, usually doctor used to give not glucose, that is ringer lactate buffer, doctor used to give patients to make them survive. So we are also treating ringer lactate buffer by plasma for medical applications, such as antibacterial effect by the plasma-treated ringer-lactate buffer, plasma-treated water, and plasma-treated other buffers like PBS or other physiological saline solutions. In agriculture cases, we are working that how plasma is affecting their like proteins and metabolites. So for this, we are targeting proteomics and metabolomic approach to check whether plasma is doing really a wonderful effect on the agriculture part or not. Seed germination, plant growth, already reported several times by us, many papers published. That is old history. But nowadays we are checking whether we can control metabolites of plasmas or various kind of proteins of plasma, which are responsible for immune system of plants. So this is a new topic, immune system of plant, not human being, immune system of plant we are targeting. So we can have many kind of stress even to the plants by pathogen, pest attack, that is, and we can have two kind of stress, that is abiotic stress. Abiotic stress is like not by the biological material, it is by like salt solution, salt concentration, or some presence of heavy metals, etc., etc. And biotic stress is by like pathogen, pest, or those. So those kind of defense mechanism we are checking in the plant whether we can control by using plasma or not. So this work is ongoing, and we got a recently very wonderful result. Many genes related to abiotic stress and biotic stress proteins are altered by our treatment. And we also checked by the metabolomic approach that the many proteins related to energy production inside the plant are enhanced by the plasma as compared to other non-important genes. So this is a future perspective. We are working on various virus inactivation strategies by using plasma. We are working on immunomodulations. We are working on the green synthesis of nanomaterials and green sustainable agriculture. We are working on standardization of doses for medical applications. And most important is commercialization. Commercialization is the most important point here that we are working on. And I will show you some slide on commercialization also after this slide. So these are commercial product we have. You can see this is the dental device for dental cleaning. You can use this device for the dental problems. And this is based on the jet plasma I showed you earlier. And this device is available by our collaborative company, that is Bioplatec. So Bioplatec is selling this device in Korea, in China, Indonesia, Philippines, Malaysia, even in Singapore and Thailand. Maybe in India, they will start their programs. They will start their brand soon. And another device is PlaBio. This is device by PlaBio for skin and aesthetic. That is for specifically for related to makeups and the people who have wrinkles, the people who have skin problems, they can use this device to make them beautiful. And another device is for wound and burn healing. That is also based on plasma. So if because it is very hard to treat wounds related to burn, and usually people died within few weeks because drugs, the chemical drug cannot control properly those wound made by the burn. So we developed this device specifically for those burn patients to treat them significant, efficiently. And another is Plasma BT. This is by Solin Medicare. And this is also for skin disorders. Many kind of pimples or acne can be removed by this kind of two devices. So these are all available in market, specifically in Korea and nearby region. You can say East Asia. And this is our last year device. We developed it and put in the market. Already 5,000 devices already sold in the market. In China, in India, we sold out some devices, not many. Thailand, Malaysia, Singapore, and specifically in South Korea and Japan, we sold out up to 5,000 devices of coronavirus. This is coronavirus inactivation and fine dust removal device by using plasma. We say simply plasma air sterilizer. for hospitals, home, and public facilities. So we can use this device, simple device. It is in my home also. You can see in my home, the fish are enjoying this device very well. And continuously its position is here, and I never found any fish died because of presence of this plasma device. So our collaborative company recently got many kind of award from Korea government to develop this kind of coronavirus inactivation device by using plasma. So these are collaborators. These are my 4 PhD students: Manoma Negi from Himachal Pradesh, Shweta Borkar from Maharashtra, Paritosh Patel from Odisha, and Apurva Jaiswal from Uttar Pradesh. So these are 4 students who are working under my guidance as a PhD students, and we have many master course student and undergraduate student in research also. And these are our collaborative members: Professor Inha Choi from Korea, and many other people we have in collaboration. Even some Indian professors we have collaboration. He is my professor supervisor, PhD supervisor, Ramesh Chandra. He was Vice Chancellor of Bundelkhand University, as well as he was director of ACBR in Delhi University. And nowadays he is professor of Nuclear Material Center in Delhi University, Nuclear Medicine Center, Nanomaterial Medicine Center in Delhi University. Professor Akhilesh, Professor Jagopal from DTU. Rajiv Varsne sir, he is director of DRDO. He was also my supervisor in India during my PhD course. And other my friends and colleagues, all are, I used to thank all these people in my presentation. Thank you very much. If you have any question, you can ask me now.
Thank you, sir, for this informative session. Sir, am I audible?
Yes, yes.
Yes, sir. I was one of those people who believed that plasma medicine is basically about blood plasma, not the fourth state of matter. So
yeah, many people used to confuse by plasma, like blood plasma.
So this session was really informative in that matter, sir. Also because we are studying nanobiotechnology this semester, and previous semester we studied immunology, so we were able to connect. I can speak on behalf of my students, on my classmates, that we can connect better. We could connect better to the presentation of yours. Also,
yes.
the one thing that fascinated me the most was the non-thermal plasma therapy as compared to the radioactive therapy, because people suffer more by the radiation therapy than cancer itself. So this is so futuristic and really interesting.
Yes.
So sir, and to conclude, I would like to request Dr. K.D. Rawat sir to give the vote of thanks.
Thank you, Nitya. I welcome Dr. Nagendra Kaushik. He provided very interaction session. All the aspects are covered in his lecture. They show plasma medicine use, past, present, and future. So I think it is very dynamic lecture, and I hope all the students will take benefit from this lecture. So I present my sincere thanks to Dr. Nagendra Kaushik and Director Training and Placement Cell Pratap Singh. So he provided opportunity to all the students to maintain their dynamic personality by interacting training and placement cell. And I would like also thanks to Professor Neeraj Dilbagi. He provided the opportunity to all the students, and especially Nitya Arora. I also present my sincere thanks to Nitya Arora. She also working in a good and dynamic work in the training and placement cell. So I hope all the students will interact in future to Dr. Nagendra Kaushik, and one of them or two of them, couple of them are in future student of Dr. Nagendra Kaushik. So thank you very much, Dr. Nagendra.
Thank you, sir. Thank you very much.
Nitya.
Thank you, sir. Thank you so much, sir. Now I would request everyone to please open their video cameras for a brief moment so that we can take a group picture. Please everybody open their video cameras. And Sneha, please make sure that you take a picture. Sneha? Am I audible? Am I audible?
Yes.
Yes.
Yes, audible.
Is the picture taken? Sneha? Okay, so pictures have been taken, I think. So thank you everybody who has joined the webinar. This concludes the webinar of today's session. Thank you so much.
Yes. Yes. Thank you, everyone.
Thank you, sir.
Thank you, Dr. Nagendra, once again.
Thank you, sir.
Very informative session was.
Oh, thank you. Meet you later again.
Yes.
Bye, everyone.
Hello, Nitya.